Hello, this is Caitlin Hall at SoftGenetics, and I would like to welcome you to the Mutation Surveyor webinar, Working with Your Mutation Surveyor project in the Graphical Analysis Display. This is a three-part webinar series pertaining to how to use the Graphical Analysis Display to review and edit your project data. This is the first of the three webinars, and today we will review the different components of this feature. Please view the follow-up webinars to learn about the functionalities of the Graphical Analysis Display in more detail. The Graphical Analysis Display, commonly referred to as the GAD, is a principal feature within the Mutation Surveyor software that provides visual representation of sample analysis. By displaying the results of the patented anti-correlation technique in the same view as the locations for both genomic and gene locations, users can easily confirm, add, and edit mutation calls. The GAD is a location of the variation tracks used to track the annotation from any external databases that have been imported, as well as from your user knowledge database, and also provides exclusive access to certain reports. In this webinar, we will be taking a look at each pane of the GAD from top to bottom in detail. But before doing so, please make sure you are familiar with loading samples and running a project. I would recommend viewing the introductory overview webinar if you're not yet familiar with the basic functionalities of the software. The first pane at the top is a nucleotide pane. This pane is designed to show the base call alignment between the GenBank reference and sample files. The information within the loaded GenBank file, along with the selected mutation project settings, will determine what and how information is displayed here. For example, under the default project settings, chromosome coordinate information, when included in the GenBank file, will be displayed in the position information at the top of the pane, and the nucleotides of the GenBank file will be oriented in the gene direction. With these default settings selected, we will see alignments to negative strand genes indicated by the upside-down nucleotide lettering in the GenBank file string and the ordering of the chromosome coordinate numbering decreasing from left to right. Capitalization of the GenBank nucleotide letters indicates positions within a coding exon. The nucleotide sequence of the GenBank file will always be displayed as the top sequence. When working with bidirectional data, the ordering of the remaining sequence will be that of the forward reference trace file, forward sample trace file, reverse sample trace file, and reverse reference trace file. Color-coded asterisks flag positions where traces differ in base call. Sequence differences between reference trace and sample trace files are marked with black asterisks, and red asterisks indicate sequence differences between the GenBank file and reference trace files. Please keep in mind that Mutation Surveyor does not rely on base calling for mutation detection, so the placement of an asterisk does not necessarily mean a variant is called at this location. Located directly below the nucleotide pane is the amino acid pane, and this pane is arranged to display the one-letter or three-letter amino acid abbreviation directly below the middle nucleotide of the codon. The top line is the translation taken from the GenBank file, and the line below is that from the sample. Variants that result in a change in amino acid will be displayed in the bottom line, and the mutation project settings include options to display changes resulting from indels, such as frame shift variants. Relative gene location can be determined by viewing the amino acid position located along the top of the pane or using the cDNA index position provided along the bottom of the pane. Just like the nucleotide pane, the strand location for the gene and the selected project settings determine the direction of translation. If we zoom out, we can also see the CDS number provided within the brackets of the exon. The tracks pane is an optional feature and will only be displayed if a whole human genome has been imported along with annotation databases and/or the creation of your known deletion tracks, and the chromosome coordinates are available for the given project. Queried tracks will be displayed in the pane with green tick marks indicating positions of reported variants. Hover over the tick mark to display the available annotation for the database. This feature comes with its own settings that can be used to filter project variants and/or include specific annotation fields in project reports. To learn more about the variation tracks, please view our webinar titled User Knowledge Database with External Annotation. The Sample Traces pane, also known as the electropherogram pane, displays the traces for a selected sample. When working with bidirectional samples, The traces will be grouped by direction. Traces to the right of the blue vertical bar represent the forward direction, and traces to the right of the tan vertical bar represent the reverse direction. The outermost traces are the reference traces, with the sample traces appearing in the middle. The innermost traces are the mutation traces. Trace type, meaning reference or sample, name, sequence direction, lane quality, and any trace comments are displayed above each trace. Please note that reference traces derived by the software, which occurs when no reference traces are loaded into the project, include the word synthesis in the file name. This pane is discussed in more detail during Part 2 of this webinar series. The pane located at the very bottom of the GAD is the mutation table pane. This pane displays all of the mutation call information for the selected sample. The standard blue and red text color coding applies here, as well as the pink and purple background coloring. Additional color coding is included for bidirectional analysis when a call is made in only one direction. This color coding will follow the vertical lines in the sample traces pane, with the blue background color indicating a call made only in the forward directional trace and the tan background color indicating a call made only in the reverse directional trace. This pane may be used to edit, confirm, or delete any mutation call, as well as to add any comments. Adding false positives and artifacts to the user knowledge database, viewing actions performed by the software and users, and accessing a mutation call's audit trail can all be accomplished through the mutation table pane. Finally, double-clicking the line of a mutation call in the table updates all of the panes above to this location. Please view Part 3 of this webinar series to learn more how this pane can be used in conjunction with the sample traces pane to add and edit variant calls. Each pane on its own provides different information that can be helpful for variant review, and when used together, they provide a comprehensive view at any location. Specialized icons located in the top toolbar help customize and navigate through the display of the GAD. A panel of 4 icons located along the top controls which panes of the GAD to display in order to view only the information pertinent to your analysis. To the right of this panel is the Show Lines icon, used to filter for only desired peak colors. A section is included for the 4 main peak colors in your sample trace and reference trace files, and another section lists the mutation peak color options and their associated nucleotide change. Next, the Zoom In and Zoom Out buttons can be used to gradually expand or restrict the field of view. The Display Mutations with 1D or 2D settings icon toggles the GAD between 1D and 2D views in accordance to your threshold set on the Two Directions tab of the Mutation Project settings. If any bases have been edited, an icon to reprocess modified files will be present in the toolbar. Select this icon to reprocess the samples using the base edited sample or samples. The Binoculars icon opens a search bar that can be used to search within the GenBank file. Queries can be entered using either position or sequence, and sequences can be reverse complemented if needed. Finally, 2 graphical reporting options become available only when working within the GAD: the GAD report and the export GAD report. The GAD report displays the graphical analysis display for all of the sample trace files within the same config together in a linear view. Preferences can be set to include all of the samples in the config or only those containing mutation calls. The export GAD exports a single mutation call per page or per file, depending on the selected output option. In this report, the config information and variant call information are accompanied by a snapshot of the GAD centered on the particular mutation call. This concludes Part 1 of our webinar on utilizing the graphical analysis display in Mutation Surveyor software. For more information or to request a free 35-day trial, please visit softgenetics.com or send an email to info@softgenetics.com. All technical inquiries can be sent via email to our technical support group at tech_support@softgenetics.com. Thank you for joining me, and please check the description of this video for links to all of the related webinars referenced today.